So this is an opportunity for us to really think about natural products and medicinal chemistry, and specifically the way that it relates to metabolism. So if you think about this in the big picture, we've been looking at things like the citric acid cycle, right? And so part of what we need to recognize, though, is that there are many other aspects of metabolism that we have not yet explored. And so those are really shown here. And I show this not to overwhelm you, but to really give you a sense of the complexity, the nuance, and the beauty that is found in metabolism holistically. So if you were to go and study, say, for example, in biochemistry, you would look at these cycles in much more depth. And so while we've explored the citric acid cycle and we've thought about glycolysis and we thought about beta oxidation, the reality is that's just dipping your foot in the enormous ocean of metabolic reactions that are possible. So we're going to look a little bit at the different types of molecules and the types of reactions that you can think about. So natural products as a whole tend to be grouped either according to structural features or according to where they come from. So what genus of their plant source. So for example, these are some examples of floral chemical compounds and the different flowers that they come from. Notice if you were to just examine this, you would say that structurally they're very similar. In fact, there are very few differences between these chemical compounds. So natural products tend to be grouped either according to a structural feature, right, or to something that they come from. So these are examples of flavones. So we have flavone, isoflavone, and neoflavone. Notice, right, these are all isomers of each other. A secondary metabolite, then, is a small molecule that is not essential to the growth and development of the producing organism, and it is not classified by structure. So here are some examples of secondary metabolites: prostaglandin E1, morphine, erythromycin A, benzopenicillin. And these natural products are all things that are going to come from this specific pathway. So DNA to RNA to proteins to then small molecules or natural products. So for example, the odor of a freshly crushed mint leaf, like many plant odors, is due to the presence in the plant of volatile C10 and C15 compounds, which are called terpenes. You actually looked at terpenes when you were in lab. And so while this odor, right, is common to terpenes, it's actually found in a variety of plants. So alpha-pinene, linalool, beta-caryophyllene, myrcene, limonene. These are all examples of different chemical compounds found in different plants, but that all contain terpenes. And just in case you were wondering how many terpenes there are, there's a variety, and you're going to notice that they're all built from this kind of common intermediate. So for example, myrcene, ocimene, limonene, sabinine, alpha-pinene, camphene. These are all examples of compounds that are built out of this chemical structure, which this is, right, the basis for. If we think about another class of chemical compounds or natural products, so the term steroid applies to compounds that contain a hydrogenated cyclopentaphenanthrene carbon skeleton. So cholesterol is an unsaturated alcohol, the principal constituent of human gallstones, as well as many other places where cholesterol is found. It's notorious for hardening arteries, but actually cholesterol, either free or in the form of esters, is widely distributed in the body, particularly in nerve and brain tissue, of which it makes up about 1/6 of the dry weight. And here's what's really interesting. You could pursue a career in graduate school studying cholesterol because the function of cholesterol in the body is not well understood. So if we take a look here, there are several different examples. So ergosterol, stigmasterol, estrone, vitamin D2, equilenin, and progesterone are just a couple of examples of the types of steroid compounds. Here are some others: testosterone, endosterone, cortisone, digitoxygenin, digitogenin, and ecdysone. These are all, again, derivatives of steroids. If we look at another class of natural products, the term alkaloid or alkali-like is commonly used to designate basic heterocyclic nitrogenous compounds of plant origin that are physiologically active. So here are some examples of how these compounds are made. In this case, they're made from the amino acid tyrosine, and these are synthetic conversions. So we convert tyrosine to dopamine And then we have several intermediates on the way to making things like themane, codeine, and morphine. When we think about natural products, one of the structures that comes to mind are antibiotics. So the first antibiotics of medicinal value were discovered by Alexander Fleming in 1929. These were metabolites of the microorganism Penicillium nototurn. And really, it's this urgent need for non-toxic antibiotics that was recognized during World War II and resulted in a team effort of English and American scientists to develop really efficient methods for preparing penicillin by fermentation, and to really think about the clinical and chemical studies that would result in an understanding of how this molecule works. So the structure is unusual in that it has a four-membered cyclic amide ring. A beta-lactam is what that's called. And it was the first example to be discovered of a natural product with this type of ring structure. And so fermentation can produce penicillins that differ only in the nature of the side chain group R. And the common natural penicillin is penicillin 6, in which R is the phenylmethyl or the benzyl. So the cephalosporins are antibiotics produced by the bacterial strain Cephalosporium, and they are closely related to the penicillins. Thus, cephalosporin C has a beta-lactam ring, but a six-membered sulfur-containing ring. And there are a variety of classes of antibiotics. So when we think about natural products, one of the things that's important for us to recognize is that there are several different types of beta-lactams, just like there are several different types of aminoglycosides, or glycopeptides, or quinolones, or oxazolinones, or sulfonamides, or tetracyclines, or macrolides, or ansamycins, or streptogrammins, or lipopeptides. And these are all examples of bacteriostatic agents or bactericidal agents. So notice there's a difference here. Bacteriostatic agents restrict the growth and reproduction of bacteria, whereas bactericidal agents cause bacterial cell death. Other examples of natural products are the prostaglandins. These are found in low concentrations distributed in a large number of organs, tissues, and the body fluids of mammals. Their precise biological role is not entirely clear, but they are known to induce strong contractions of smooth muscle tissue in the lungs and uterus, and they also lower blood pressure and sodium levels. So the prostaglandins have been implicated in the control of the pituitary hormones released from the hypothalamus and the incidence of pain as a response to fever and inflammation. So the analgesic property of aspirin possibly may result from the inhibition of prostaglandin biosynthesis. So here are some examples of prostaglandins and their variety of structures. So here's the point. When we think about medicinal chemistry, before the 1800s, there was a variety of use of plants, plant extracts, inorganic material for medicinal purposes. Around 1805, morphine was isolated from opium. Its structure was determined in 1935 and its synthesis in 1952. In 1828 was the first organic synthesis. That was the synthesis of urea. And then in the 1840s to the 1850s was the first synthesis of organic compounds used in medicine, such as chloroform or ether for the purposes of anesthesia. And there were several examples of early synthetic drugs. So chloral hydrate is a sleeping pill, acetylsalicylic acid, phenazone, benzocaine, protonsil. These are all examples up until the early 1900s. If we think about medicinal chemistry today, these are the top 10 drugs in 2014 and their sales in the millions. If we think about that today, and we think about those numbers in 2017, you can start to get a picture of the types of diseases that drug development is used to treat, and then the companies that are responsible for developing those drugs. So medicinal chemistry is the preparation of biologically active compounds. It's the study of their metabolism, their mode of action at a molecular level, and then the construction of structure-activity relationships, or SARs. And this is where a lead compound that has a desirable biological activity with some therapeutic relevance, but typically has some type of shortcoming that needs to be overcome through the development of analogs. So how do they determine whether or not medicinal chemistry works? Well, they look at a dose response curve. This depicts the relationship between the drug dose and the magnitude of the drug effect. So drugs can have more than one effect. They can also vary in effectiveness per patient. And so different sites of action and different affinities for receptors are critical to identify. And then the effectiveness of a drug is considered relative to its safety. This is a therapeutic index. So these are dose-response relationships for 4 analgesic agents. Each curve represents the increase in pain threshold as a function of the dose. The pain threshold is the magnitude of painful stimulus required to elicit a response. So this is the percentage of elevation in pain threshold, and this is the amount of the dose. So a good way to think about this is the dose that's required to reduce someone's pain experience. What's critical to identify here is that not all people respond to a similar dose of a drug in a similar manner. So this variability can be based upon individual differences and is associated with toxicity. This variability is thought to be caused by things like the pharmacokinetic factors, which contribute to differing concentrations of the drug in a target area, pharmacodynamic factors, which contribute to differing physiological responses to the same drug concentration, and then genetically determined allergic or immunologically sensitized responses. When we think about the binding of these compounds, it's really important for us to identify that there are several different modes of binding. So you have a target site and a neuron receptor. You can have inactive storage deposits, such as in bone or fat. You can have the absorption and distribution through membranes or the gastrointestinal tract or the peritoneum, skin, muscles, or lungs. You also have differences in terms of drug administration, and then you have the functions of things like the liver, blood plasma, plasma protein binding, and metabolites in terms of transporting those drugs. And then you have excretion itself through the intestines, kidneys, lungs, and sweat glands, and the excretion products. Part of what I hope you're starting to realize is that the absorption of drugs can occur in a variety of places, whether that's in the stomach or the small intestine, through oral drugs, as well as injection, and then this distribution through the bloodstream to the liver or to the kidneys. And so one of the things we want to explore is something called Lipinski's rule, which states that in general, an orally active drug has no more than one violation of the following criteria. It has no more than 5 hydrogen bond donors, it has no more than 10 hydrogen bond acceptors, and it has a molecular mass less than 500 Daltons, and it has an octanol water partition coefficient, or log P, not greater than 5. Note that all of the numbers are multiples of 5, which is the origin of the rule's name, Lipinski's rule of 5. So it's critical here to note that molecular mass or molar mass are used in stoichiometry calculations in chemistry. In related terms, another unit of mass often used is the Dalton, or unified atomic mass unit, which when describing atomic masses and molecular masses is defined to be 1/12 of the mass of one atom of carbon, or in older works is also abbreviated as AMU. So hydrophobic drugs with high octanol water partition coefficients are preferentially distributed to the hydrophobic compartments, such as the lipid bilayers of cells. Hydrophilic drugs with low octanol water partition coefficients are then found in the aqueous compartments, such as blood. So let's take a look. Here's aspirin. It has a molecular weight of 180 Daltons. It has 1 hydrogen bond donor. Can you identify it? It has 4 hydrogen bond acceptors. Can you identify those? It has a C log P, or the octanol water partition coefficient, of 2.44. Here's another example. Artemisinin has a molecular weight of 282 Daltons. It has no hydrogen bond donors. It has 5 hydrogen bond acceptors, and it has a C log P of 4.15. You will have several examples to identify in terms of Lipinski's rule of 5. But this is what you want to start practicing, is to thinking about, right, the rules specified. So no more than 5 hydrogen bond donors, no more than 10 hydrogen bond acceptors, molecular mass less than 500 Daltons, and an octanol water partition coefficient not greater than 5. So if we go back here, right, each of these still meets Lipinski's rule. The last thing we're going to talk about in terms of medicinal chemistry are this idea of isosterism. So these are compounds or groups of atoms that have the same number of atoms and electrons. So examples would be N2 and CO, or N2O and CO2, or N3 and NCO. This is what we refer to as Grimm's hydride displacement law, which is the addition of a hydride to an atom gives the resulting pseudoatom the properties of the atom with the next highest atomic number. So this is the hydride displacement law. This idea of isomorphism, isosterism, and covalence is something that was originally identified by Langmuir in 1919. The idea is the following. So bioisosteres are structural moieties with broadly similar shape and function. The function should be biological, but it can sometimes modulate other properties. And then bioisosteric replacement is the replacement of functional groups. So molecular scaffolds are a subset of bioisosterism. And it's the idea that we want to identify the core functional or structural element and then make adjustments as we see fit. So next time we'll do a quick review of metabolism in general and some of the big ideas. Just as a quick recap, remember, here's keto, the transition state, and an enol. This is something that's going to show up again and again, particularly on the final. All right, thanks so much. Have a great day.